we talk about biologic, we need to understand this is an application from recombinant DNA technology. Now, when the DNA and we can add in the cell, the cell, what they will do? Cell will either copies or translates. There will be production of protein. So, DNA piece into some specific protein, and that protein we called as biologic medicine. So, it could be different insulin analogs, interferons, erythropoietin, and nowadays we are using many monoclonal antibodies. So, all they are biologics. So, as we had discussed, biologics are large protein that based on some specific protein moiety derived by the injecting DNA into some specific cells, the cell will produce that protein. So, when we talk about the size, they are very large molecule. So, from small molecule to biologics, the basic difference is in the size. So, if we start from an example of a small molecule, aspirin, which is only 180 Dalton size. Now, when we move towards biologics, starting from the simple biologics we have, we know many of us use daily, that is insulin. So, they are the very simple biologic. So, they are only 5700 Daltons. But if we increase the complexity of the biologics, then what we get? We get growth hormone, almost 191 amino acids and comprise with 22,000 Daltons. And when we talk about monoclonal antibodies, they are huge therapeutic protein. So, they have almost 1300 amino acids and the size is 150,000 Daltons. So, from left side you can see the small molecule, the complexity increased, we will get insulin, growth hormone and then monoclonal antibodies. So, biologics are large protein based therapeutic that vary in size and complexity. Not all biologics are same. So, according to the size, according to the amino acid sequence, they become more complex. So, let us understand how they differ from a chemical based medicine. Already we had discussed the small molecule drugs, their size is much lesser. Then we get some small biologics like growth hormone, then the large biologic like monoclonal antibodies. Insulin are smaller than the human growth hormone. So, we can say they are the smallest biologics. If we discuss insulin, then growth hormone, then monoclonal antibody, insulin is the smallest biologic among these three, whereas monoclonal antibody is containing more than 25,000 atoms and a huge molecule. So, the difference is when we talk about biologic medicines, they are extremely large, small molecules, as name suggests, they are very small. The structure is complex here. So, increasing complexity, we will get more complex biologics, whereas the simple molecule medicines, they are very simple in nature. They are unstable, this is biologic, they are not that stable, they are highly sensitive to temperature and pH. So, that make them more difficult to characterize and produce on a large scale. So, the chemical structure variability is also there, high degree of variability, whereas the stability was more in favor with small molecule medicines, and there is low degree or very less degree of chemical structure variability. And as we can understand the complexity of the biologics, they should be used through the parenteral route. Oral, it is not possible. Whereas the small molecule, they can be used through any route of administration. Immunogenicity is a huge issue with biologics that we need to understand, because they are therapeutic protein, so they will evoke the immune response inside us, the antigen-antibody reaction. So we need to understand, because this anti-drug antibody which will be produced by this biologic insertion inside our system may result in reduced efficacy, also may arise some safety concerns. But for small molecule medicine, there is very little potential to produce immunogenic response. When we will discuss about biologic, we need to know about this monoclonal antibodies, antibody drug conjugates, a new concept, peptide bodies. So this peptide bodies or peptide bodies, they are peptide and some FC portion of that antibody. So FC fusion products. So one part is peptide and another part is FC fusion. So ultimately they will produce a product. So we will discuss a little bit about that, the cytokines and some growth factors. So when we discuss biologics, we need to know about the simple biologics first, that is insulin. Here we are talking about simple biologics, how they can be prepared. Now you can assume after this discussion the complexity of production of large biologics, how they are complex. So first of all, the DNA sequence need to be chosen. The differences in DNA sequence that may result in variations of the final product, that recombinant protein product. Then next step is cloning. So difference in the expression system can lead to variations in the level of recombinant protein expression and their stability. So this cloning is a very important step. Next is cultivation. So difference in growth conditions result in variations in the recombinant protein and their byproducts. Then protein production, that therapeutic protein production. So even the cell lines are different, that may lead to some variations in terms of post-translational modification of the recombinant protein. Purification is again an important step. If any difference happened here, this purification process, then there will be some variation in the final product, and that may cause some increased aggregate formation, and that may result in some toxicity. Finally, the formulation. Even there is some difference in the formulation that may influence the final product.